Thank you for everyone for joining us today for this second webinar that we are hosting by the Functional Genomics Screening Lab. I'm Chun-Hao Wong. I'm the Muna lead for the FGSL team.
I'm Uli Kunze. I'm the AstraZeneca lead for the FGSL lab. And we want to quickly introduce what the Functional Genomics Screening Lab is about. This new lab is a joint venture of the Medical Research Council, AstraZeneca, and the Nanotherapeutics Institute here at the University of Cambridge. We focus on arrayed CRISPR screening and complex models, and will form academic and industry partnerships to identify new targets in biology and development and disease. We run a call for project proposals constantly and review proposals twice a year. So academics can apply with their idea for an arrayed CRISPR screen. The next deadline is on the 3rd of November. And if you're interested in applying, please scan the QR code that you can see on the slide and you get to our FGSL website with more information. To mention that successful applicants will get fully funded projects and they will be executed here at the FGSL in Cambridge.
Great. So we as a group are very interested in applying functional genomics tools to actually systematically tease out genetic factors underlying often very complex phenotype. And we also hope to use this webinar series as a forum to encourage conversation and also knowledge exchange between the community and around these kind of areas that are actively being investigated. Our webinars are typically recorded and posted on our page so that you can review them later as well. And I have a link here that I've just posted on the chat that you can go to visit our previous webinar on the FGSL that we give more in-depth introduction. We'll move on to the talk and then to any questions that you can submit in the chat sessions. And this leads me quite nicely to introduce our guest speaker today for our second webinar. So we're delighted to welcome Delphine Larrieu. Delphine is a principal investigator at the Altos Lab, Cambridge Institute of Science. Prior to joining the Altos Labs, Delphine was a Wellcome Trust Henry Dales Fellow and an assistant professor at the University of Cambridge. Her research investigates the mechanism underlying nuclear envelope dysfunction with aging and premature aging in progeria syndromes. Her talk today will describe a recent genome-wide array CRISPR screens in cells derived from progeria patients to identify targets that might reverse this tough age-related nuclear envelope phenotypes. And I believe this is a great example of how unbiased screening in a highly relevant model can drive further hypothesis generation. And with that, I'll pass to Delphine, and we'll look forward to the talk.
Thank you so much for this nice introduction and for the invitation to discuss our work. So yeah, today I will discuss about how we used whole genome arrayed screening to identify new mechanisms and targets in the context of progeria syndromes. So I have to disclose my conflict of interest. I am currently an employee of Altos Labs. However, everything I will present today is the result of the work of my previous lab that was carried out at the University of Cambridge. So when I was at the university, the main focus of my lab was to understand nuclear envelope regulation and links with disease. And before I get started into giving you more details about the work, I need to introduce you to the nuclear envelope. So most of you might already know what the nuclear envelope is, but for those of you who don't, the nuclear envelope is actually a very important structure that is crucial to maintain the architecture of the nucleus, but also the architecture of the chromatin. And you can see on this schematic that it is also contiguous with the endoplasmic reticulum. And if we zoom in on this structure, you can see that it is composed by many different types of proteins. So on the inner nuclear membrane side is a network of intermediate filaments of lamine proteins called the nuclear lamina. And this nuclear lamina is really very important for not only for maintaining the integrity of the nucleus, but also for organizing the chromatin inside of the nucleus, because this is the site of anchorage for heterochromatin and for telomeric chromatin. So through this role, it also regulates gene expression. And this function is also, some other proteins also contribute to this function. So many proteins that directly interact with the nuclear lamina. And these include hemerin and BAF that I will mention again later in this talk. You can also see at the nuclear envelope proteins of the nuclear pore complex, obviously involved in transport of proteins and RNAs between the nucleus and the cytoplasm. And we also have transmembrane proteins that connect the nuclear interior to the cytoskeleton and play very important functions for transmitting mechanical forces from the outside of the cell into the nucleus. So you can see that, with all these different functions, the nuclear envelope is really crucial for maintaining cellular homeostasis as a whole, really. And so somehow it's not very surprising that mutations in nuclear envelope proteins cause very dramatic diseases. And among them are progeria syndromes or premature aging syndromes that are caused by mutations in specific nuclear envelope proteins. So the classic progeria syndrome called Hutchinson-Gilford progeria, or HDPS, that I'm sure most of you have heard of, is caused by a mutation in one of these nuclear lamina proteins. And you can see how dramatically this mutation affects the structure of the nuclear envelope and of the nucleus. And at the level of the patients, it leads to very dramatic accelerated aging features. The average lifespan of these patients is around 14 years old, and they usually die from cardiac failure. More recently, a mutation in a different type of nuclear envelope protein called BAF, which stands for barrier to autointegration factor, was identified as being the cause of another progeria syndrome called Nestor-Guillermo progeria, or NGPS. You can see again how the mutation in this BAF protein strongly affects the structure of the nuclear envelope and of the nucleus. And similarly, these patients present, accelerated aging. But interestingly, the tissues being affected by the mutation are very different from the ones affected in the case of the laminae mutation. So here, while mainly the cardiovascular function is strongly affected, in Nestor-Guillermo progeria, the cardiac function is absolutely fine, but instead they experience very strong skeletal abnormalities. So this NGPS and the BAF mutation will actually be the focus of today's talk. Just to give you a little bit more insights into this NGPS syndrome. So it's actually caused by an autosomal recessive mutation in BAF. So it's an alanine to threonine mutation that leads to early onset symptoms. So the children usually start displaying signs of aging around the age of 2 to 3 years old. However, compared to the classic progeria, the clinical progression is much slower. So the lifespan is longer, they live over 30 years. And as I mentioned before, they mainly experience very severe skeletal abnormalities. You can see one of the patients with very, very severe scoliosis. They also experience bone resorption, osteolysis, as well as some phenotypes that resemble normal aging, such as osteoporosis, joint stiffness, lipodystrophy, or hair loss. And as it is the case, unfortunately, for a lot of these very rare disorders, there are currently no cure or no treatment available. So a little bit more insight into BAF itself. So BAF is actually a very small protein, but it's an essential protein, so you can't engineer BAF knockout. It is lethal. And this is because it's involved in crucial cellular processes such as nuclear envelope reformation after mitosis. It can also recognize viral DNA upon viral infection. It has been involved in the regulation of transcription, as well as in the maintenance of nuclear integrity. And all these roles are being performed through the interaction of BAF with different types of proteins including nuclear envelope proteins, lamine A and hemerin, and BAF can also directly bind to the DNA. Now, in the case of the NGPS-associated BAF mutation, before we started working on this topic, little was known about the consequences of this BAF mutation. We knew that the mutation caused again abnormal shape, abnormal nuclear envelope integrity, and that it was also associated with mislocalization of this nuclear envelope protein hemerin from the envelope into the cytoplasm. And our work, as well as other people's work in the past couple of years, have identified that the mutation actually prevents the proper binding of BAF to lamine A at the nuclear envelope. And that prevents repair of nuclear envelope ruptures that we observe in these patients. So one of the questions that we had when we decided to start working on NGPS was, how could we find new ways to restore nuclear envelope integrity in NGPS cells? So we managed to obtain fibroblasts from 2 different patients that I will call here NGPS1 and NGPS2. You can see again the strong nuclear envelope abnormalities in these 2 different patients. And the goal was to be able to take an unbiased approach and therefore a whole genome approach to find new ways to rescue these defects. And there were basically 2 aspects to this screen. The first one was to gain insights into the biology of BAF, understand the consequences of the mutation better, but also obviously identify potential new therapeutic targets for NGPS. So I will spend a little bit of time to explain what setting up the screening actually involved, because it was, as you can imagine, quite a lot of work. And most of you might be more familiar with the sort of, I call it classic pooled flow cytometry-based CRISPR screens, which nowadays are pretty quick to set up. But in our case, these nuclear envelope phenotypes could really only be observed by microscopy. So, we couldn't possibly use flow cytometry as readouts of these nuclear envelope defects. So we had to turn to the microscopy-based screenings. And I just want to mention that, it was the very early days of these CRISPR-based arrayed screens. So there was basically no precedent in the literature. So we had to literally, set up everything without any basis. The first step, and I think it's probably the most important for any of you planning a screening approach, is to really think about the cellular model you want to use and to really characterize in depth the phenotypes that you want to investigate in your screen. So in our case, we wanted to work with fibroblasts from NGPS patients. This was obviously quite complex because we observed some differences in the phenotypes between the different patients. And obviously this can be due to patient heterogeneity, as it is the case, we all have a different genetic background. And to be able to really identify the phenotypes that were directly caused by the BAF mutation, we decided to establish isogenic cell lines by taking these NGPS patient cells and reversing the homozygous mutation using CRISPR-Cas9. So this way we had isogenic cell lines, NGPS corrected, as I will call it later in the talk, and we still had the wild-type cells, from an unaffected individual that we also use as a basis for comparison. I will tell you a little bit more about the phenotypes we used in the screen in the end, but we spent a lot of time to really, identify specific phenotypes of these NGPS cells. Then obviously you have to find and validate the combination of antibodies that you want to use to, to look at these specific phenotypes and to develop high throughput analysis pipelines. So this is just to show you how the reversion of the mutation looked like by looking at the sequencing. So this is the wild-type cell line that we use as a basis, the NGPS cell line with the BAF mutation, And this is the corrected cell line where we reverse the mutation with CRISPR-Cas9. Now, the second step was to have, to obtain these NGPS fibroblasts with stable Cas9 expression for the CRISPR screening approach. So, that involved basically identifying the best promoter to express Cas9 in a homogeneous way. And initially, we had planned to use a polyclonal population of cells expressing Cas9. Unfortunately, despite the fact that most of the cells were expressing Cas9, we had high level of heterogeneity of knockout efficiency from one cell to the next. And when working with these arrayed screens, you can't afford that. So we had to, in the end, establish single cell clones and select, clones with the highest, efficiency of knockout. These are just examples of specific clones. But we also didn't want to use one single clone for the screen, because then you can, end up with results that are due to, clonal effects. So to prevent this problem, we decided to pool 3 different clones together. And that was our NGPS Cas9 population that we used for the whole genome screen. The next step was to test the 2 different systems that were available at the time. So that was in 2018. So yeah, it might seem like a long time ago now. But at this point, there were only 2 companies offering these whole genome arrayed libraries. One of them was Horizon Discovery, offering a system based on CRISPR tracer RNA. So it's basically 2 components, 2 separate components to form a functional guide RNA. And Syntego, that was offering the single guide RNA library. Just to keep in mind that the cost of these libraries were really high. So for the CRISPR tracer RNA, it was 70,000 pounds. Sorry, whereas for the Syntego library, it was 100,000 pounds. So we really wanted to, to identify what was the best system that worked, in our specific cell model. And again, this might be very different depending on the cell type you are using. So we tested, both approaches using immunofluorescence readouts as well as Western blotting readouts. But in both cases, we had very similar efficiency of knockdown. We confirmed, similar efficiencies using a mini-targeted screen also for experimental validation, where we basically obtained a cherry-pick library of mitotic genes. And this was to basically have easy readouts of cell growth using an IncuCyte system. And you can see that, again, we had pretty similar effects of, using both systems, where knocking out these mitotic genes led to a strong reduction in cell growth. So based on the cost, we decided to go for the cheapest one out of the 2, which was the Horizon CRISPR tracer RNA library. So we were then finally ready to set up our primary genome-wide screen. So the library was composed by 4 different CRISPR RNAs against each single human gene arrayed in 384 well plates. So to cover the whole genome, we were working with 61 384 well plates. And in each plate, we could include positive and negative controls for quality control. We then used a robotic liquid handling machine to plate our NGPS Cas9 cells together with our tracer RNA. And we incubated the cells for 3 days, incubated our antibodies, and then analyzed basically imaged the plates with a high throughput microscope. Once primary hits were identified, we had to set up a validation screen. And this time, this was performed in triplicate. As you can imagine, the primary screen could not be performed in triplicate, but the validation screen was done in triplicate. And there we decided to deconvolute the CRISPR RNA. So we had 3 individual CRISPR RNAs against each of the hits arrayed in wells. And we did some hit validations and follow-up that I will show you in a minute. So the whole thing took us about 5 years from start to finish. It was a huge amount of work. We also had the COVID lockdown in between that didn't really help. And basically another year and a half for publication. So this is a good timing to present this work because it was finally just accepted in Nature Communications. So this is how it felt really. It's there are no unrealistic goals, only unrealistic deadlines. We never would have imagined that it would, it would take us so long. But obviously, when you have to set up everything yourself, I mean, this is quite a big piece of work. Just an overview of the high throughput platform that we used in our screen. So we were able to use a SciBio Felix liquid handling robot in the institute next door. And this is the high throughput microscope that we had available at the CIMR at the time, that came with the quite powerful image analysis software that we could use for the screen. Right, so going back to the first step, what were the phenotypes that we used in the screen? So the first one is, the very obvious one that I presented at the start is the nuclear shape. I mean, this is one of the hallmarks of progeria. This is the easiest to look at using, some nuclear envelope stainings, but even potentially just a DAPI stain. That was very nicely picked up by the automated software. And this is just to show how to show you how the quantification looks like, again, between the wild type cell line, the NGPS cell line, and the corrected cell line. The second phenotype, which was again something that was reported before in the literature for these NGPS cells, is this mislocalization of nuclear hemarine from the nuclear envelope into the cytoplasm. So again, that's something that you can pick up with this software. You can basically discriminate the nucleus with your nuclear stain from your cytoplasm and then quantify how much protein you have inside or outside of the nucleus. And this is again how the quantification looks like. The third phenotype was something that we discovered in my lab in parallel when working with these cells, where we saw basically that these NGPS cells experience a lot of nuclear envelope ruptures during interphase. And again, we spent quite a lot of time to be able to pick up these ruptures and quantify them properly. And this can be identified by some kind of bleb of the nuclear envelope that end up rupturing over time. So you can see again how the software was able to pick up the nuclear contour and the bleb, and how the quantification looks like. The final phenotype that we included is the presence of micronuclei that we observed in these cells that can be a marker of genomic instability. And as the name indicates, these are pretty tricky to pick up in a high throughput manner because they are really, really tiny. But luckily, the marker that we used to pick up the nuclear envelope ruptures was able to really nicely also identify these micronuclei. So, we decided to combine these four phenotypes, and we wanted to identify genes that, when being knocked out by CRISPR-Cas9, would be able to normalize or rescue at least 3 out of these 4 nuclear envelope phenotypes. The reason behind that is that we really wanted to narrow down the number of hits to the ones that would be the most powerful in acting across different nuclear envelope phenotypes. Obviously, we have the list for each single readout, but then you end up with quite a big number of genes, and then you don't really know where to start. So having this combination of phenotypes is also one of the the powers of these microscopy-based screens, is that you can really combine a number of phenotypes in your specific system and obtain potentially more specific targets. So with this approach, the primary screen identified 130 hits. So you can see that already even in the primary screen, the number of hits was quite small out of the 20,000 human genes. So it was a manageable number of genes to work with for the validation screen. And oops, I'm missing a slide. Anyway, in the validation screen, we were able to confirm 43 hits out of the 130 genes. with very high confidence, meaning genes that were basically present in the 3 replicates of this validation screen. This is the list of the 43 hits, and you might be surprised to find genes that have no obvious connection to the nuclear envelope. Again, this is, the power of the screens is that, you end up with hits that you could have never hypothesized. So, very few genes with known connection to the nuclear envelope, nuclear transport, and nuclear pore. But the strongest gene enrichment was actually genes associated with translation, which was a bit of a surprise. So the first question we asked was, why would translation inhibition be beneficial to these NGPS cells? So, basically it was translation genes that, when being knocked out by CRISPR-Cas9, would improve these nuclear envelope phenotypes. So, nothing obvious there. So to address this question, we decided to first look at, what was the basal level of protein synthesis in these NGPS cells. And to address this, we took a click chemistry-based approach where we incorporate in cells a methionine analog that you can tag with a fluorescence tag. And then when you look at, the fluorescence intensity, it somehow reflects the rate of incorporation, so the total protein synthesis in the cells. And with this approach, we could see that the NGPS cells basically had higher levels of basal protein synthesis. The same occurred when we overexpress the BAF812T mutant in a different cell background. That was basically enough to increase protein synthesis as well. And interestingly, we could see that when we used, standard protein synthesis inhibitors such as cyclohexamide or silvestrol, we were also able to improve nuclear envelope phenotypes of these NGPS cells. So it really seems to be, something to do with the protein synthesis rate. And interestingly, when we look at how downregulation of the 43 hits we identified in the screen can modulate protein synthesis rate in these NGPS cells, we observe that in most cases, the knockout of these genes, even the ones that don't have direct known involvement in protein synthesis, were able to somehow reduce the rate of protein synthesis. So we think at this stage that, this protein synthesis reduction and sort of rebalancing of protein synthesis rate might be a common mechanism by which knockout of the hits can rescue the nuclear envelope phenotypes in NGPS. In addition to this change in the rate of protein synthesis, we also identified increased errors during protein synthesis in these cells. And this is interesting in regards to what happens in physiological aging, because this is something that has been observed in normal aging and that contributes to proteostasis imbalance. And recently, there are a couple of papers, including this one, that have reported the effects of genome-wide error-prone translation or protein synthesis in mice in vivo. And you can see that basically when you induce errors during protein synthesis in mice, the mice display premature aging features. And actually, these features somehow resemble what is seen in these NGPS patients. So the mice show skeletal abnormalities, chest deformities, this very strong hunchback posture, as well as hair loss, lipodystrophy, and early death. So potentially, this could be a direct cause of some of the phenotypes observed in NGPS patients. And this screen may have identified potentially a new link between nuclear envelope integrity, protein synthesis, and longevity, at least in the context of this model. We confirmed some of the hits in vivo using a C. elegans model with a collaborator at the University of Sevilla in Spain, where they established this NGPS worm model that show a shorter lifespan, as well as very similar nuclear abnormalities as we observed in patients, as well as deregulation of protein synthesis as well. And in this model, we could also see that deletion of some of the hits from the screen, including some of the ribosomal proteins, were able to rescue the viability of these worms. So with that, this is my last slide. The next step would be obviously to pursue target validation and potential therapeutic development. But I hope this has highlighted the power of this microscopy-based screen to identify new biology and potentially new targets. But I just want to reiterate that there are really critical points when you think about setting up these types of screening, including really thinking hard about the relevance of the cell model you want to use. Make sure that in your specific system and the specific CRISPR platform you will use, you have really high efficiency of gene knockout in a homogeneous manner. And again, spend a lot of time to make sure that your phenotypes are highly reproducible and quantifiable. And I would recommend, whenever you can, to combine phenotypes to really limit the number of hits. So, in this case, if we were to pursue this work, the next step could be screening small molecule libraries against some of the hits. So some of the hits are actually druggable targets, or potentially develop new tools to target the hit for degradation, for example, through PROTAC-based approaches. And with that, I will finish to thank everyone here for joining the seminar. Thanks for the invitation. Sophia Breusegem was a postdoc in my lab, who was the main person involved in this screen. Together with Anne Janssen, we had a lot of help from facilities, and we could access a high throughput robot in the institute next door. And obviously, I want to thank all the collaborators and the funding bodies involved in this study.